You are tuned in to Relativity.
Wait, are we live? We're live. Let's get real.
Relativity. REALITIVITY. Relativity. Hey, Petra, how's it going? Welcome back to episode 2 of our CRISPR investigation here.
Yeah, it's great to be back with you. I'm excited to talk about CRISPR and its discovery in the natural world, and then our transforming of CRISPR into a tool for human gene editing, all kinds of gene editing, actually.
We'll get into the first part here of the technology, if you can use that word, the how bacteria came about this through their own methods of fighting infections of their own. It's really it's akin to our immune system, but quite a bit different. I wanted to start off a little bit different here, Petra. I got a couple of questions, a little bit more on the philosophical side, but I think it's going to tie in greatly to the discussion that we're going to have. And so what my first one is, I would kind of be curious as to what is the number of sort of biologists at the upper levels of PhD level of people doing research there are across this world. And the reason I'm bringing that up is because I want to do a comparison against that number. And I just think that we have the number. I know when I was going to school and I was looking, what could I do in this field? There were not a lot of jobs. And so I kind of want to compare that to what is the number of people graduating with, say, technology degrees and software and electrical engineering and everything like that, hoping to get jobs at Google or someplace else in Silicon Valley.
You know, it's a great question, and I don't have an answer on how many biologists there are, in across the globe, but I'm pretty sure you're onto something, which is that there's this kind of allure of the tech jobs, and I love technology too, so it's not like I'm looking down on the tech jobs. But what's so amazing about the CRISPR story, and to tie it back into the philosophical part of your question, is that through and through, it's a curiosity-driven journey that ends up creating this huge revolution in kind of gene editing and what we're able to do with the genome of not only ourselves, but also of plants and food and animals and all other kinds of creatures. So the whole story throughout, which we'll get into when we get into that, is about people just studying things they were interested in because they were interested in them, because they wanted to find out what the truth was, because they were just curious, right? It wasn't about, I have to reach this quota or I have to develop this algorithm to drive this number of sales. It wasn't that kind of outcome driven field that we think of as a lot of science and a lot of technology. And there's nothing wrong with having an outcome and a goal. But when your goal is just discovery, you don't know what you're going to uncover. And that's kind of the beauty of CRISPR. And I think a lot of biology is that if you know what you're looking for, you might miss a lot of really beautiful, unbelievable things that you kind of discard as unrelated to your goal. So that wasn't really an answer to your question, but I really love the question.
I wasn't expecting to hit you up with a bunch of numbers and have those right away. We could probably do some Google searches and put that in the show notes. But one of the things I wanted to get to, and I think I'm hopeful because you brought up something about curiosity, and I read Walter Isaacson's book on Jennifer Doudna, who will play a very important role in our discussions today. And it talked about her as a young kid in Hawaii and her just general curiosity. There was so much nature around her. She just was a very inquisitive young kid, and that carried out throughout the rest of her life. And it leads to discoveries like that. And one of the other things I want to say, it's not just curiosity, it's creativity. There's all these people, that person's a creative. Well, I actually believe that everybody is a creative in their own way. If you think about the stay-at-home mom, she's creative in everything that she does all day long of how to do this, get the kids to soccer, pick up the groceries, do all these different things. And you can be creative in any job that you're doing. I think software coders are some of the most creative individuals that we have out there. Yes, there's probably factories of coders that are just programming basics, but when they're developing algorithms and they're searching for new ways to do that, that's a very creative process. And I don't think many people would ever put coders into the bucket of creative people. And so kind of tying back in here to my original question is I love that the curiosity aspect and the creativity of a lot of these people mean that we're going to have some very top-notch people into biology fields. Let's just call it the biochemistry, biology, genetics, virology, immunology, that type of study. And it's great because we're going to get some of the best and the brightest in there, but the numbers I think are still going to be low. And so when we start talking about things, you brought up the topic of in silico the other day. So how in the world Google AlphaGo is going to now tackle the protein folding problem, right? But how do we know that the computer got those proteins
AlphaFold.
folded? Yeah, how do we know that they're going to get those right? Who's doing the check and verification that those proteins, what the computer came up with, is correct? And the amount of proteins they're going to be able to do and kind of have this stockpile sitting there waiting for these curious and creative individuals to go and do something with is going to be immense. So I have no idea how they're going to know that. Where's the biologist sitting in that lab verifying all these proteins that are going to get folded?
It's a great question. And I think you might have hit on another topic that we're going to have to do a show on, which would be trust in AI, which I find an absolutely fascinating topic because we're entrusting more and more really important functions of our society, from determining if you get bail to determining if you get a loan to potentially creating the mechanisms for folding for proteins, and how do we validate what that AI is telling us, right? It's a huge question, and it gets even more tricky when you get into quantum, where we don't even have the capacities necessarily to validate what the quantum computer is telling us. So I love it. We're going to have to write that one down, Mitch. That's going to have to be a future topic.
Okay. And so right as we get into the story of CRISPR, I want to bring up one last thing. So it took a very long time for CRISPR, kind of from when they first started seeing these, the CRISPR, you can say what the acronym stands for. I always mess it up. But when they first found this in E. coli, it was in the late 80s. And it wasn't until the 2011-12 timeframe that, stuff really started to happen with it. And we get to where we are today. So there's been about 30 years in time. Now, I go back and look at this, and I go to the number of biologists and related individuals in this field, and I can look at the number of papers that are produced and think about the number of genomes that we're now getting documented and cataloged. And we're getting more and more information across the board on all these different bacteria and viruses and other animal genomes and everything else. And there's this wealth of data just sitting there. Where are all the people that have that curiosity? Where do they have the time and the bandwidth to go through and do what happened with CRISPR in that area? And again, we'll tie this back to AI. Maybe there's going to be a way to make AI somewhat curious and be able to help us out in this area. But I think there's gold mines out there. And I don't necessarily mean gold mines in making money. I just mean gold mines of wealth and information and new knowledge and things that can help us in the future, just waiting for us, and all these papers that have been written. And it just needs that one person to say, that doesn't make sense to me. Why does it work that way? Anyway, we better get on topic here, Petra.
We are on topic. This is fantastic because that is the story of CRISPR, right? So I'll try to spit it out here again. Clustered regularly interspaced short palindromic repeats, or CRISPR. And you mentioned it briefly there, Mitch, but the first team to actually discover these repeats was a team at Osaka University in Japan in 1987, led by Yoshizumi Ishino. And apologies if I said that name wrong and all of the other names wrong that I will say later. But they were looking at the gut microbe E. coli, as you mentioned, and they published a sequence of a gene called IAP. And they were trying to understand how that gene works. They were sequencing some of the DNA surrounding it, hoping to find the spots where the proteins landed to turn that gene IAP on and off. But instead of that switch that they were looking for, right, they had that goal, but they also had the curiosity to notice something that didn't fit into what they were trying to discover at all. So they were finding these 5 identical segments of DNA. And if you remember from our last part of this series on CRISPR, DNA is made up of those building blocks that are called bases, right? So the rungs of the double helix ladder, if you will. And they kept finding these 5 segments that were each composed of the same 29 bases. And then those repeating sequences were separated by blocks of other DNA called spacers, which each had a unique sequence, unlike the identical segments. So they were kind of looking at them like these DNA sandwiches and had no idea what they meant. They had never seen anything like this before. And like you said, this was back at a time where sequencing sequencing DNA was a really, really difficult process. So it wasn't like they could just go and kind of compare it to other kind of microbes and see if this had been seen before. So they kind of wrote it up in their paper. I mean, they didn't have the answer to what these gene sandwiches were doing. But I love that they kind of finished their paper with the quote that says that the biological significance of these sequences is not known. So they didn't know what it did, but they knew enough to record it. And I just love that was kind of the preface to this whole journey to discover CRISPR and what it does in cells and how we can use it for our gene editing purposes.
I think real quickly we should talk about those clustered repeats just a bit. That is actually where the palindromic part of the whole CRISPR wording comes in. So each one of those repeats was actually a palindrome, right? So reads forward, backwards, the same. And so that was pretty interesting, I think, to them as well. And I think one of the things to point out, Petra, you hit on it, is, they in the 80s and up into the early 90s, it was pretty labor intensive to pull out, to look at someone's genetic code and be able to separate it and to be able to, kind of get that classic ATC, GGG, CAG code may say. There was you had to put these on these gels and it took a long time. And, it took a while to make any progress and map this type of stuff out. Not to mention that there's millions of things that you could potentially map out. So slow process. And like many things, it was kind of like, oh, let's put that on the shelf and maybe technology will catch up and help us out here. I like to think of this little section as like, do you remember those jump ropes back in elementary school? They were the plastic ones with the red and white stripes on them, so it was just spacers like that,
Yes,
right?
absolutely.
Yeah, so like each of the white ones might be the spacers that we're talking about. And so I'm sure you'll jump into now what those spacers are made up of. I think that's pretty interesting how this little system works. And this is the technology. This is what's going to blow everybody's mind, I think, Petra, is that I don't know if people can get in their heads that it's bacteria. It's this little cell wall. There's nothing much to it. It's just got a few organelles and things in there, and it's just this little cell, and it comes up with this technology we're about to discuss. So it blows my mind.
It is really cool. It is really cool. You have to remember the conversation about eukaryotes and prokaryotes, right? So prokaryotes are bacteria and archaea, which tend actually they factor into this discussion. They're really small, a lot smaller than eukaryotic cells. And eukaryotic cells are what we are made up of, and they contain those membrane-bound organelles like a nucleus and mitochondria. So think of a prokaryotic cell as a lot simpler, and yet they have evolved these unbelievable capabilities that kind of mimic our own immune system and that are so unbelievably efficient at what they do at protecting the cells. And we tend to think of ourselves as obviously very superior beings with our intelligence and all of our unique immune systems and other systems that make humans who we are. But it turns out that bacteria have quite a lot of intelligence themselves in kind of this way that they've evolved to fight viruses. But we're getting ahead of ourselves, right? Because we don't even know at this point, we're in the 80s, they don't even know what these little gene sandwiches are doing, or these jump rope gene sequences. And I think the next kind of development that happened, at least in the research that I did before this, is looking at how in the 1990s, as we talked about, all those technical advances allowed all these scientists to speed up their sequencing. And they kept seeing that these strange genetic jump ropes were appearing in a staggering number of different species of microbes. So they eventually became so common that even though scientists didn't yet know what they were, they needed a name, right? Because at that point, they're trying to figure out what they're doing. So that's where the name CRISPR came from. I believe it was a collaboration between Ruud Janssen in the Netherlands and also Francisco Mojica, who's a Spanish scientist. And they started noticing these genes everywhere, started to figure out what they actually did in bacteria.
And so to me, this is another just amazing miracle that we have going on here, right? They are in this eon long fight with viruses and bacteriophages to battle one another, right? And this is a defense system that they have come up with. And here we are talking about, you mentioned how small they are, but you think of how small a eukaryote versus a prokaryotic cell, I just went the wrong way. A prokaryotic cell is to a eukaryotic cell. We have spent eons fighting both bacteria and viruses ourselves with our immune system. And so it's so interesting to see how each of these different systems have evolved over time to get to a point where I would almost say we live in some level of symbiosis, right? It's not any one of the three are overtaking any of the others. We struggle on a day to day basis to fight bacteria and viruses all the time. Anyway, I got us sidetracked again, Petra, as I tend to do. I think we were going to get in.
I think it's fantastic.
I think one of the things that we can start here on, if we want to talk about the technology a bit, is the way that a virus will come in and try to infect a bacterial cell is basically it attaches to the outside and it shoots a little bit of its own DNA into the cell. And the idea with that is that DNA is going to get in there and disrupt the bacteria cell. But this is where the CRISPR system, and I think I called it a caller ID system the other day, is able to recognize this, what do you want to call it? This unwanted
signature.
This signature of an unwanted piece of DNA entering where it shouldn't be.
Yeah, absolutely. So to kind of parallel that with this discovery process that's going along here. So I mentioned Janssen, who was part of the team that named CRISPR, and they noticed that CRISPR was always accompanied by this collection of other genes, which they called CAS genes, CAS genes, short for CRISPR-associated genes. And these genes encoded these enzymes, right, that could cut DNA. But no one at this point could say why they did so. And this is in the early 2000s. So they could see, okay, there's CRISPR sequences. They're accompanied by these Cas genes. And we know that these Cas genes can cut DNA, but they don't know yet why they can cut them. And then there was the next kind of step in this amazing journey, this saga, the CRISPR saga, is that this team led by Eugene Koonin at the National Center for Biotechnology Information in Maryland, he noticed that these CRISPR spacers, so those individual unique jump rope segments in between the palindromic segments, looked a lot like the DNA of viruses. So there are kind of three teams now who are entering. It's never, as we talked about, it's never one scientist making a breakthrough. It's all these different people working on different aspects of a problem. Anyway, these three teams started to notice that microbes were using CRISPR as a weapon against viruses for exactly the reason that you said, which is that the virus would attack the bacteria, and the bacteria would take a snippet or a signature of the virus's DNA and then insert that DNA into its own DNA. And in that way, it would be able to recognize when a viral intruder that it had seen before was trying to enter and impair the cell. So it's really fascinating. I saw one article that called Molecular Most Wanted List,
That's
which I just love. I think it's so good. Yeah.
Our genetic jump rope is better, though. And Petra, I want to stop on and talk a little bit about CAS, and it played a very important role in the saga that was the development of this CRISPR technology, right? One of the big debates goes back and Jennifer Doudna and Emmanuelle Charpentier. Did I say the French correctly? I hope I did. If I didn't, I apologize.
Beautiful.
They teamed up together for quite some time. They've been a team for almost all of this, but they had their rough patches, and there was many people along the way that also were contributing to this. And with anything, anybody kind of wants a little bit of credit for their work, right? There was a point in time where they hadn't associated this Cas protein with everything else that was needed to do it. And it turned out there was about 3 or 4 different papers. They were all coming out. And again, I got to talk about, this was one of those crazy stories. And with every advancement, there was always like 3 or 4 papers all coming out at a very similar time. It was like, wow, we found this. And they all kind of went and did their own thing, but they came back to something very similar, and they each approached it in a different way. And it either confirmed or didn't confirm and led people down different paths. But this whole Cas protein was one of those areas. And in fact, it's still to this day, there's, lawsuits out to who actually found this technology. I think some friendships were maybe battered a little bit over, which paper was it, who thought of it first, even going down to the point of people looking through the notebooks and the dates in their scientific notebooks to figure out this whole, who really recognized that CAS had to be there, or Cas9 specifically had to be there for this to occur. It's fascinating. And I'm going to encourage everybody to go get Walter Isaacson's book. I'll try not to repeat too much out of that because I think you should get it. I think you should get any of his books. They're all phenomenal. But this one is very good, and it really goes into the details. He does a great job of trying to represent each of the parties, if you will, from their perspective on who found what and when. And it was crazy, Petra. It was so fast, too. It was literally like this group would find something and this group would take the next step, and it was moving extremely fast for science, for bureaucracies.
Sure. Another cool kind of part of this story, and I'm just fascinated by the kind of stories and in all of this. So I'm gonna keep dropping them in here. But did you read about the microbiologist? I'm gonna totally destroy his name. It's Rodolphe Barangu. I'm so sorry, Rodolphe. You are amazing. But he was making at Danisco, the big yogurt maker.
Oh yes, the yogurt.
Yeah, this is at the time when Kunin and those other scientists that you talked about were kind of hypothesizing that the microbes were using CRISPR as kind of a weapon against these viral invaders. There wasn't enough really known to prove it at the point. Rodolphe Barangu, who was working at Danisco, was thinking about this issue because it wasn't just interesting to him. It was obviously fascinating, but it was also important for his employer, who, Danisco and yogurt makers depend on bacteria to turn milk into yogurt. So if you lose an entire culture to an outbreak of a virus that kills your bacteria, you're going to have a problem, right? So Kunin is suggesting that bacteria could use CRISPR against these bacteria-killing viruses. And so he actually tested it. So he tested, there's a milk fermenting microbe called Streptococcus thermophilus, I think is the right one. He infected it with 2 strains of viruses. So the viruses killed many of the bacteria, but some of them survived. And when those resistant bacteria eventually multiplied, their descendants of those parent bacteria turned out to be resistant too. So it's clearly a genetic change had occurred. So they found that the bacteria had stuffed DNA fragments from each of those 2 viruses into those spacers, those protospacers between the CRISPR genetic jump rope segments, those unique parts. And this is the cool part, when the scientists cut out those spacers that were mimicking the virus's DNA, the bacteria were no longer resistant to that virus. So it was kind of I believe it was, correct me if I'm wrong, I believe it was the first proof that that's how the bacteria were using CRISPR. And the cool part about it is because he was at a major manufacturer, Danisco, those manufacturers immediately started to select for those sequences in bacteria. So they weren't using CRISPR, the tool, yet as a human tool, but they were basically using the natural incidence of CRISPR in order to create bacteria and products that could withstand virus outbreaks. So he said he's moved on. I think he's a professor somewhere now, but he said that if you've eaten yogurt or cheese recently, then you've probably already eaten CRISPRized cells, because this was a huge development for the yogurt and the food and the cheese kind of industry to learn how to protect your food from these viruses. Anyway, sorry, that was a total diversion, but I think it's fascinating, all the different pieces that came together here.
Well, what I find fascinating about that is I know the first thing that I start thinking about when I think of CRISPR and it's where is it going to get implemented in, I'm thinking of medical type stuff, right? Whether it's diseases or whether it's cosmology or something like that, right? Those are the first things that come to my mind. And at no point in time would I have, jumped to this. Oh yeah, the yogurt industry, cheese industries, and there's probably many other areas where this is going to be important. Yeast, for the bread industry, yeast for beer, right? I actually saw something the other day that somebody had made some fluorescent beer, and I think it came from introducing fluorescence into the yeast. Again, finding stuff and looking at how other people have been creative and curious enough to pull it into other fields and how it's going to work. I guess in this case, it was another field that actually kind of tied a bow around the whole idea of what that CAS gene was doing and getting CRISPR to take that next step. So good talk there on that. So let's see, Petra, we owe a little bit more description on how this works. And we were just getting into the CAS protein and what the importance of it in this process, as where I think we left off.
Yeah, absolutely. So I guess we've arrived at Jennifer Doudna and her team, right? They really figured out that's where they really figured out that the microbe can use viral DNA to basically target those CAS enzymes into, think of them like precision guided munitions at this point. But she and her team, Jennifer Doudna and her team, really saw that these CAS enzymes were programmable, right? Their purpose is to cut DNA, and they're programmable. And she looked at that with her team and said, "Oh, this is a tool." And to me, before we jump into this, and I'm sorry, we will jump into this, but to me, it was so fascinating because you can look at, and again, we've touched on the difference in complexity in terms of a bacterial cell compared to a human cell, right? Or compared to a plant cell, or compared to an animal cell. And it's not immediately obvious that a microbial tool like this would be applicable to human DNA, right? The eukaryotic and prokaryotic kind of cell difference, right? And yet she looked at it whereas others were kind of keeping it in this domain of bacteria, which makes total sense. She kind of looked at it and saw it as a tool, not just for bacteria, but for all kinds of different forms of life that we encounter. And I think that's just such a cool jump.
It's pretty noble too, to some of her first thoughts were, how can I use this to cure some of these just gnarly diseases, genetic diseases that we have. I do want to hit on one thing here. And, there was a big step of going from how the bacteria are using this and sort of the machination, if you will, of all of this. And so in the bacteria, you had this little bit of CRISPR RNA, and it had the spacer, and it had the genetic information that the Cas protein would come back around, and then that's how it would grab that the DNA from the virus and the bacteriophage. And there was also a little bit of tracer RNA. They had to do a little bit of genetic modifications just on that little process alone, to where that they had basically, and there was a gap in that tracer RNA to the CRISPR RNA, so they had to close that gap. And then they had to bring in what's known as the guide RNA. And once they had done that, that's how they were able to basically target any genetic sequence that they wanted to. So they would open this up, and then as you open up, so as that Cas protein would sort of go around the DNA, open it up, and then what it would do is it start replicating, make a new piece of RNA, and then, or that RNA would get in there and start to replicate, and then the cell would just come in and fix itself, right, like it should. And so it would just make the other strand, the complementary strand in all this. That is a very difficult process and takes a lot of experiences and a lot of specialized tools and equipment to pull that off. So I like the idea that CRISPR sounds very simple, very elegant in the way that it's put forth in the bacterial system, but it took these folks a lot of work, and they had graduate students across the globe working on this to come up with and be able to make this technology work in such a way that they could target cells and insert that DNA strand. Very complex.
Absolutely. And I love what you brought up there. So I mentioned Francisco Mojica briefly, who's a professor in Spain, and he was one of the people who actually named CRISPR, that delightful acronym there. And he said in 2012, when Jennifer Doudna, there were several teams working on this, right? But in 2012, when he saw that they had basically used Cas9 to program a pair of genetic scissors to cut that DNA strand, he said it shocked him. It really surprised him, because he's a microbiologist of prokaryotes, right? Of those simple, really simple cells. And when you cut the DNA of a bacteria, of a prokaryotic cell, you kill it. But what he didn't know, because he was in the world of prokaryotic cells, is that in eukaryotes, when you cut the DNA, you actually also activate a repair mechanism, right? Yes. I believe it's called homologous recombination. So that opens a possibility to actually rewrite the DNA. So instead of just cutting and killing whatever target DNA, you also create the opportunity to really reprogram that, because basically so picture those CRISPR is often referred to as genetic scissors. We'll have to come up with a better name than that, Mitch. But anyway, so it's often referred to as genetic scissors. It cuts the DNA, and then other DNA that's in the vicinity that has a matching end basically to the two recently cut ends will be able to recombine with it and kind of heal that strand. So that creates the opportunity not just to cut or turn off DNA, but also to repair it in ways that we find important, valuable, health creating or disease preventing or otherwise interesting.
Well, one of the things that I want to hit on there is, again, this is going to go back to the breadth of the team that it took, but it's also going to go into the curiosity and creativity of these individuals that were working on that, right? So if your day just deals with these prokaryotes, you're going to know that when I cut the DNA and it tries to go in and come back together, there's a mutation and it kills it. And that's exactly the attack vector from the viruses, right? But if in a eukaryotic system, as you mentioned, we have these repair systems. And so I find that the thing with PhDs, whether it's in chemistry or biology or physics or whatever the case may be, they are so highly specialized. Yes, they have many, many years of work and knowledge across the board in their field, but they get so highly specialized that I don't know, maybe the blinders on at some point in time. And it took someone like Jennifer Doudna and that team to say, oh wow, while it kills these things in bacteria, I know that we've got a repair mechanism in place in our animal cells, our cells. This has potential. And they went for it. So, Petra, we've taken a look at how this works, and there are plenty of videos and everything else. You can go to YouTube and find great stuff on, these great, whether those whiteboards, where people draw on the whiteboard and it shows everything. And I highly encourage everyone to go out there and look at that. But I want to take a step back and just talk about gene editing in general. This is nothing new. It's gotten easier. This is going to democratize sort of the application of gene editing. But when you think about it, we've been doing this since the beginning of time. Look at our dogs.
Absolutely. I would say our tools have only gotten better. And I'm going to have to find one of my favorite examples to point to about this very topic is teosinte, which is kind of the ancestral source of modern corn. And it literally looks like a strand of wheat. Like you would never think that it's related to those beautiful golden ears of corn that you get in the summer. But it looks like a strand of wheat. And basically our ancestors bred this ancestral corn, barely even corn, this ancestral plant and turned it into these juicy kernels of corn. And you're absolutely right. It's as old as time basically is our transformation of the environment around us to better suit our needs. And I would say just that our tools have gotten better over time, right? Our tools have gotten better. And the cool part about CRISPR, well, one of the many cool parts, let's be honest, but it's not even the first time that scientists have borrowed tricks from microbes to build a tool for gene manipulation, right? It's not even the first instance of that. Okay, I'll tell you, and then maybe we might have to back up because I'm going to jump a few millennia here.
Okay.
But so some microbes, defend themselves from these invasions by viruses using molecules that are called restriction enzymes. So this was actually one of the kind of motivators behind the rise of the modern biotechnology industry in the 70s. But there's something to kind of clarify here. So there's two types of immunity, right? There's the innate immunity that we're born with, which protects us and is not really unique to our environments. And then there's the adaptive immunity. You can think of like your antibodies, right? So if you get sick with something, then your body will make these antibodies. And so the purpose being to recognize that same pathogen and defend against it. So it turns out that bacteria have a similar kind of innate immunity as well as CRISPR, which you can think of as an adaptive immunity, right? Because it's learning about the signatures of those viral invaders. So restriction enzymes, to go back to why I originally brought this up, they actually chop up any DNA that isn't protected by these kind of shields. So microbes have these shields that really protect their own genes, and then they basically attack whatever unshielded DNA there is in terms of viruses and other parasites. So in the 1970s, when molecular biologists first figured out how to use these restriction enzymes to cut DNA, it was it was a revolutionary advancement in that time as well. But the problem with these restriction enzymes is that they don't have this precise cutting ability that CRISPR has, right? So they basically just shred any foreign DNA that's not protected by those shields. So as a result, it's really hard to control. It's hard to control where those enzymes cut DNA and where they don't. Basically, the CRISPR, the CAS system, right, has actually evolved to create that kind of targeted control over which DNA gets cut. And that's why it's part of the reason why it's an adaptive immunity, right? Because you learn as a microbe. I love just the image of microbes learning. But anyway, they learn from kind of those viral invaders. Here's their DNA signature. Here's their caller ID, as you said, and here's how we can defend against it. So restriction enzymes are just kind of a one size fits all defense system. Anyway, how's that for a tangent, Mitch?
I love our tangents. That's a good one right there. So one of the things that we need to kind of get into is talk a little bit about what are some of the uses, what are some of the things that are coming up, sort of in the future, kind of set us up for episode 3 just a little bit. One of the things that I think we can get this discussion going with is, as of right now, there are places where if you take your eggs and your sperm, you can actually look and see, or you can actually get an embryo going. That'll be this comment will get us into hot water with some folks, right? But you can go through all those embryos or all those eggs and sperm and kind of look for genetic markers that are of value for what you want, right? And so you could actually go through and look for things. Once we get a better understanding, maybe it's you can pull out Alzheimer's. You can look for height. You can look for, say, an athlete that has speed versus endurance, or endurance over speed, or eye color, or just whatever in the world you want. And that's sort of the designer baby. And that's possible now, right? You can pretty much go through, as we talked about, the genome is now mapped. We know what most of the genes do. There are still some interactions in between genes and proteins and all this stuff that probably still a bit of a mystery. But we could actually go off and do designer babies sort of in a, I don't know if that's brute force, but it's a slower and it's a less precise method, right? But what's interesting is now that we got this capability, we can actually start designing people or animals or food crops or anything else exactly how we want. So now you go to the store and you see stuff with GMO written on the side. I would argue, does the yogurt company need to put a GMO on their label for their yogurt that's just basically using a natural method of protecting itself? I guess that's a pretty bad analogy, but the corn that we've bred, the trees that we've bred to
Yes,
be more
right.
domesticated, the dogs they're domesticated, all of this is GMOs, right? So anyway.
Absolutely. Yeah, your dog is definitely GMO. Doesn't look anything like a wolf.
Not
Probably. Very
I
cool.
think one of the things too to be thinking about, and as we prep for episode 3, is this idea of biohacking. And one of the guys that we did not talk about today, I think there's probably 2 people we'll talk about in the next episode, but Josiah Zahner is one of those. And he was an individual that he really wants to get this out and make biology and science more democratized for the people, to the point of, I'm sitting here looking at his website. It's called theodin.com. You can buy CRISPR kits on this. And now you're only doing things with bacteria. And one of the cool things that he does is you got a way to take some form of bacteria. I actually don't know which strain of bacteria this is. And then you add jellyfish bacteria to it, and you will get fluorescent bacteria. And so that's, again, mentioned the fluorescent beer earlier. It's the same. You can do the yeast in the same way. We need to talk about that a little bit. We need to talk about the Chinese scientist that actually did this at the embryonic level. And supposedly there are 2 babies that were born edited to, they have prevention against HIV, which is a very noble thing, but it definitely stepped on the lines of what others believe has gone too far. So we'll need to talk about the ethics and the moral compass, if you will, of where all this is going. But I also got to talk about is this going to be a pharmaceutical thing? Is it only done in certain labs? Can we do it as, Josiah is trying to make this easily accessible to all? And he's got several kits on this thing. I might just buy one of these kits and play around with it. Looks like it's a lot of fun. It's $169 for a CRISPR kit.
Awesome.
$900 for a whole
Yeah,
genetic
you're
engineering home lab. It's pretty cool, and that's affordable. That's entrance for many of us. Now it's missing the part that I talked about, the whole how do you, you've got to be able to go off and cut a specific slice of the genome. You got to know where that is, how to cut that, put it into this CRISPR tool. That's a whole nother thing. But still, this is garage bioengineering.
Yeah, I can't wait to dive into the ethics and the questions, the pros and cons of democratized versus kind of these larger organizations controlling it all. It's a really interesting question, but I also love what you have raised about the phosphorescence and also the kind of first instance of a scientist editing cells and embryos. So real quick, just because we're still on the how CRISPR works, we kind of set this up last time, right, Mitch? There's editing cells and embryos, right, which is germline editing. And it's especially controversial because the changes that result when you edit at the germline level can be passed on to subsequent generations, right? So if you're just doing somatic cells, somatic genome editing, that will only affect the treated person that you are applying the intervention to. So a big difference there. And as you pointed out with that scientist in China who basically kind of went beyond the bounds of where CRISPR was legally supposed to be at this point. He's facing jail time as a result of that. But he had a noble goal, as you said, to make these twin baby girls, I believe, immune to HIV. But the really interesting part of that, and we'll get into the controversy, I'm sure, next time. But a really interesting part of that to me is that this part that's discussed a little bit less, which is that we don't even know if he was successful, right? Because in
Yeah.
order to know if he was successful, we would have to expose those two babies to HIV, and they've already obviously been through some questionable science already to get to this point. But that's the beauty of the phosphorescence, to tie it back in, is that the reason a lot of those early stage CRISPR experiments, or even the ones that are accessible online, like you're talking about, are using phosphorescence, because then you know for sure if your experiment was successful, right? If you show up next time I see you, Mitch, and you're glowing green, then I'm going to know that your CRISPR kit was successful.
And I'm telling you, Petra, I would do it. I mean, it would be so cool. It seems such an easy step to do that. I would literally do it. I don't know, maybe put it in my fingernails or something, Wouldn't that be cool? I would totally do it.
I would do it.
And I
Yeah.
am the problem
All
with
right,
this
let's do it.
world. That is not the bureaucratic, take a long time, do a lot of studying, set up a lot of bounds, and have government regulations and health regulations and all that around this. And I think that's one of the areas where Josiah and others are really pushing. Even Dr. Hugh, what's his last? I cannot say his last name. Anyway, we'll get that for the next one, and we'll put it in the show notes. But he's pressing the frontiers here, and you have no idea what changing that one gene, or possibly more than one gene, or it could be a sequence, it could just be a letter out of place, but we have no idea what's going to happen. So let's get real. Anytime that this is relativity, and that's what we do, right, is we get real. Anytime we go out there and monkey with nature in any way, we always get it wrong.
Absolutely, and I can't wait to dive into the topic of gene drives and the amount of what's unknown still, what still has to be discovered, and we're creating these kind of game changing new technologies, and we're unleashing them on the world, but there's that classic issue of unintended consequences, and you won't ever really understand what those are until you try it. So it's no amount of theorizing sometimes is gonna show you something that you never knew to look at. And that's kind of the story of CRISPR and how it came to be. And it's also the story of how do we kind of contain CRISPR and put these boundaries around it so that we are leveraging all of the amazing potential it has to change our society, but also making sure that hopefully we have some kind of guardrails in place for those unintended, unknown consequences of our actions. And that's always the big question mark.
The unknown unknowns. Well, Petra, I think we've given
Yes.
everybody a lot of information here on how this works at a fairly decent level of depth. But again, if you want to see more, there's plenty of great videos out there on YouTube, other sources. You can read the papers. One of the things that I'll get on this soapbox at some other point in time, I dislike the idea that we can't read all the papers. They printed in many different journals, and of course, they're not free, and it costs hundreds of dollars to get subscriptions to each of these. Scientific papers should be free. I'm going to make a bumper sticker about that. But I'm hoping that we're going to inspire kids that might listen to this to dig into this, ask questions, be curious, get into biology, other sciences, and I hope that we get parents the kind of, I don't know, background and information that they can inspire their children to also go out and pursue this type of stuff, because I think it's very important. And as we've identified here, Petra, I think there's a huge gap in the biological sciences, if you will, in the future. And so hopefully we can inspire our youth to get involved with that. So till next time.
Absolutely.
All right.
Till next time. I think I want to become a CRISPR scientist. I'll work on that till the next episode. How's that?
I'm going to send you the link. You can get some of these kits from the Odin. It's there's lots of good stuff to do here. There's one, the frog genetic engineering kit. Learn to genetically modify animals. Only $2,000. So not the cheapest thing in the world, but it's not that far from it's doable.
I want those fluorescent fingernails. I want those.
I'm good. If I can figure
All right,
I'm
Mitch.
to do it. Excellent.
Good night, love Petra. It. I can't wait to see. Good night.
Bye.